Virucidal activity of Moringa A from Moringa oleifera seeds against influenza A viruses by regulating TFEB. Highlights. Moringa A was firstly isolated from Moringa oleifera lam. Moringa A is a potential antiviral compound towards IAVs. Moringa A has good anti-inflammatory activity. Moringa A inhibits the expression and nuclear transfer of transcription factor EB, TFEB, and weaken the autophagy in infected cells. Influenza A viruses, IAVs, are highly contagious pathogens infecting human and numerous animals. The viruses cause millions of infection cases and thousands of deaths every year, making IAVs a continual threat to global health. Our study demonstrated the virucidal activity of Moringa A as a new compound from Moringa oleifera seeds against IAVs. It inhibits virus replication in host cells and protects infected cells from the cytopathic effect induced by IAVs. The EC50 and EC90 values of Moringa A for IAVs were 1.27 and 5.30 μm, respectively, when raw 264.7 cells were infected at MOI of 1. The different treatment experiments revealed that Moringa A has a significant inhibitory effect on the IAVs both before and after drug addition. Moringa A was observed to decrease the inflammatory cytokines TNF-alpha, IL-6, IL-1 and IFN-beta in H5N1 infected raw 264.7 cells. Finally, Moringa A was found to inhibit the expression and nuclear transfer of the cellular protein transcription factor EB, TFEB, and weaken the autophagy in infected cells, which could be an important antiviral mechanism. Our study demonstrates Moringa A has potent antiviral activity against IAVs, which could be due to the autophagy inhibition property. Introduction. The influenza virus is the most threatening respiratory virus to human health, causing millions of people to suffer upper respiratory tract infections every year. In the past 10 years, the large-scale outbreaks of SARS, MERS, H5N1, H7N9 and other viruses have brought serious harm to human life and health. The cytokine storm triggered by influenza virus infection can cause lung inflammation and acute respiratory distress syndrome, which is an important reason for mortality. So far, the prevention and control of the influenza virus remains a global problem. However, due to the high mutation rate of influenza viruses and the occurrence of recombination between viruses, it is difficult for existing anti-influenza drugs to cope with the changing flu viruses. The M2 ion channel blockers and neuraminidase inhibitors, which are the main drugs for anti-IAVs, face drug resistance. Although the vaccine is a core drug for preventing and treating viral infections in humans, the current vaccines are not highly protective if antigenically different new strains emerge, such as the 2019 pandemic SARS-CoV-2. Therefore, it is highly desirable to develop new drugs that would have protective activity against influenza viruses. Moringa oleifera lam, also known as the drumstick tree, is a cultivated species from the genus Moringa, family Moringaceae, and the order Brassicales. It is geographically distributed across South Asia, and in particular, India, Sri Lanka, Pakistan, Bangladesh, and Afghanistan. The moringa seeds and leaves have broad uses in both the food industry and as therapeutics. Previous pharmacological investigations have shown M. oleifera has anti-inflammatory, anti-cancer, antioxidant, and anti-obesity properties, etc. M. oleifera seeds are a promising resource for both food and non-food applications, as they have both edible and medicinal value. They have been shown to be rich in nutrients such as oils, proteins, and vitamins. The M. oleifera seeds also contain numerous bioactive compounds, such as flavonoids, phenolic acids, alkaloids, isothiocyanates, and thiocarbamate, which have been shown to have good antimicrobial activity, as well as anti-tumor, anti-inflammatory, and antivirus activities. In terms of antiviral research, some reports have shown that extracts from M. oleifera leaves have good inhibitory effects on the herpes zoster virus, HIV, and hepatitis B virus, but the key active ingredients are currently unclear. In recent years, research into the M. oleifera seeds has focused on their active antiviral ingredients. Guided by antiviral activity, our team has obtained some new compounds from the Moringa seed, of which the Moringa A, MA, has been proved to have a good activity of anti-H5N1 virus. Research in this paper is a continuation of our previous work. In this paper, we aim to study the antiviral properties of MA against IAVs by regulating TFEB, an important transcription regulator of the autophagy lysosomal pathway. In recent years, the complex interplay between IAVs and host autophagy machinery has been extensively revealed, and many studies testify that the infection of IAV strains such as H1N1, H3N2, H9N2 can activate autophagy in host cells. The mechanism is reportedly involved in viral replication and immune escape. Mounting evidence has demonstrated that autophagy is an important approach for viral passage and virus particle production in host cells. TFEB is a key transcription molecule that regulates autophagy at the transcriptional level. It can promote the transcription of multiple genes in the lysosome pathway and regulate the autophagy lysosomes production and efflux. Therefore, inhibiting TFEB to block the lysosome production may be an important strategy for host against viruses. Discussion. Oleifera seeds have become a focus of research in recent years, as they have extensive pharmacological activities and abundant levels of valuable chemical components. However, there have been relatively few antiviral studies on the chemical compositions of oleifera seeds. Our team has obtained many compounds from the oleifera seeds, including MA. This paper reported that MA has good antiviral activity against H1N1 in raw 264.7 cells, as it can inhibit RBC hemolysis.